Hi, everybody, and welcome back. What we're going to be looking at today is going to be meiosis. So we've already talked about the cell cycle, we've already talked about mitosis, and now we're going to look at the final portion to this, which is going to be meiosis. Now, when it comes to meiosis, this is going to be sperm and egg, and we're going to be specifically focusing on exactly the different phases that it goes through and the different cycles we have in order to get to our end part. So when it comes to meiosis, this is going to be the formation of sperm and egg cells. So if you can think back to our last lecture, we mentioned that mitos, again, are part of your body, so they're going to be your body cells. These are going to be the other part, which is going to be meiosis or our sex cells. So meiosis is the process used to produce something known as our gametes. So gametes are going to be, again, those sex cells, or more specifically, the formation of our sperm and egg. Now, the goal for this is to make daughter cells with exactly half as many chromosomes as starting. So unlike mitosis, where we're trying to make the exact amount and the exact copy, our goal is to make half. And one of the big reasons why is because we want to make sure we're taking a sperm cell right here, which is going to have 23 chromosomes unpaired, and we're going to come together with an egg cell, again, 23 chromosomes unpaired, so we can end up, when they're fertilized, with a zygote, which is going to have 46 chromosomes or 23 pairs. So again, this is how we end up having offspring, which are going to be 50% or half one parent and then half the other parent. So one thing that's really important to understand is the differences between diploid and haploid cells. So diploid cells are going to be one with two sets of chromosomes, okay? And then haploid is going to be with a single set of chromosomes. So that's going to be very important because as we go through meiosis, we're going to be going through our cycle essentially twice. So we'll be going through PMAT1 and then kind of PMAT2 in order to get it down to haploid. Now, one way to kind of remember this is diploid starts with a D, just like double. So in this case, diploid is double the amount of haploid. And haploid starts with an H, just like half. And haploid is half of diploid. So hopefully that's one way to kind of remember what those two terms mean. Diploid, double, two sets, haploid, half, one. So let's go ahead and do that comparison now with meiosis and mitosis. So again, one thing to remember is mitosis, the goal is to have a diploid cell, in this case, two sets, again, exactly identical to the beginning cell. So over here, you can see that, remember, we have our interphase here, and then we have our prophase, metaphase, anaphase, telophase, and then finally our cytokinesis. Now, you'll notice that when it comes to meiosis, because we're trying to end up with haploid, we're going to end up going through meiosis 1. You can see prophase, metaphase, anaphase, telophase, but then it's going to go through it one more time, prophase, metaphase, anaphase, telophase, in order to get it down to that haploid cell. Again, the major difference and the major reason why we're doing this is because in meiosis, we are not dealing with our body cells, we're dealing with our sex cells. And when it comes to our sex cells, remember, we need our sperm plus our egg in order to come up with our offspring. And the reason why is because, remember, 50% or one half is going to come from one parent, one half is going to come from the other parent. We add those together, we're going to end up with 46 chromosomes. So 23 plus 23 is going to be enough to make a person, 46, in this case, our offspring. When it comes to mitosis, we are not trying to create offspring. In this case, all we're trying to do is make exact identical copies of our original cell. So in this case, we already have our 46 chromosomes, or in this case, our 23 pairs. So all we have to do is just double them, divide it out, so we can end up with the exact same amount. Again, we're trying to have two daughter cells from our original cell. So that is the major difference. In this case, mitosis, we have a diploid cell. Remember, two sets, diploid double, versus meiosis. The goal is to try to get to haploid. So that's why you have to go through it twice. One time to get it to diploid and another to get it to haploid. Okay, so again, meiosis is the formation of our haploid. And in this case, you can see that we have an N representing our haploid. Remember, haploid is only going to have one set versus our diploid, you can see, is a 2N, meaning we have two sets. Remember, diploid double, 2, haploid half of diploid, which means it's 1. So again, formation of haploid gametes. So in this case, sperm and egg cells. And when they come together, then we end up forming our diploid. So where does this happen? This is going to occur in the gonads. And in this case, it's going to be the testicles and ovaries in animals. Meiosis is going to consist of 2 nuclear divisions. Remember, meiosis 1 and then meiosis 2. So now we're going to go ahead and take a look at the different steps when it comes to meiosis and exactly what's going on with all of those chromosomes. So here are the steps of meiosis. You can see we have our meiosis 1, and in this case, the goal is to separate the pair of homologous chromosomes. You can see we still have the same steps: prophase, metaphase, anaphase, telophase, and cytokinesis. But again, you'll notice that there is a Roman numeral next to them, meaning A1. Again, for this part, we're separating them out for the first time. So in this case, we have our diploid, and then we go through meiosis 2, which is the separation of our sister chromatids, which again are going to get us down to our haploid, half of what we originally started with. And then again, you can see we have prophase, metaphase, anaphase, telophase, and cytokinesis. And again, you can see on the side here, we have a 2, which represents that it's gone through it a second time. So again, we still have our PMAT 1, as well as our cytokinesis, and then our PMAT 2, because again, it goes through it one more time, as well as cytokinesis. So you'll notice that when it goes through prophase, metaphase, anaphase, telophase, and cytokinesis, these all are going to be essentially the same, both of these together, as what we talked about in our previous lecture. So again, we're going to have the condensing of chromosomes, we're going to have the middle part, away, total, separation, all of those things. But we're going to go and take a look at these just as a quick refresher. So this is going to be prophase 1. Remember, when it comes to prophase, one of the main things we want to do is we're dissolving that nuclear envelope, which you can see over here. It's disappearing. And you can also see that our DNA here is condensing down. So you can see it's very, very stringy over here in our early phases of prophase 1. And then you can see the mid, it's become much more condensed down. So you can see instead of all this stringiness over here, it's getting much more condensed. And then finally in our late, you can see it's very, very condensed out. And you can see those iconic chromosome shapes or those X's. So again, prophase 1, exchange of genetic material occurs at the chasm called crossover and recombination. So crossing over results in recombination chromatids and increased genetic variability. When it comes to metaphase 1, mitotic spindle fibers are attached to the chromosomes. And remember, metaphase middle, you can see that in metaphase 1, they're all meeting up at that metaphase plate. So right in the middle over there. So pairs of homologous chromosomes align at the metaphase plate, and then independent assortment is going to occur here. Anaphase, remember, ana, away, so A equals away. And what's going to happen here is, again, they're going to pull away from each other, so they're going to move away from the plate in the middle. So pairs of homologous chromosomes separate, so each chromosome consists of their sister chromatids. And then mitotic spindle fibers retract chromosomes pull to the opposite poles of the cell. So again, they're going away from each other. Now, one thing that's really cool here is you can see that recombination portions here. And you can see that those crossing over points have exchanged some of those pieces. And again, the reason why is because it's all about increasing genetic variability. That's why everybody is different from each other, because again, we want that variability because it helps with survival. All right, so telophase and cytokinesis. So this is going to be the end of the first nuclear division. Remember, we're going through this once, and then we're going to go through this one more time. We're trying to get down to haploid. So again, same thing that's going to happen. Nuclear membrane is going to reform because we want everything to stay within the nucleus. Cytoplasm is going to begin to split. You can see that about right here and about right here. Each chromosome still have 2 sister chromatids, but the sister chromatids now have a different genetic information from each other. So again, this is going to be completely different because unlike mitosis, where we want exactly the same thing, in this case, we want that genetic variability. We want those differences because that is going to be important for survival. So now we have two haploid cells formed. Each cell has one set of chromosomes. But what we're going to do is we're going to go through one more time. So it's going to be pretty much exactly the same thing. Prophase 2, centrioles duplicate, move the opposite ends. Again, spindle fibers are going to come back. And then again, that membrane is, again, going to dissolve. Metaphase, again, they're going to do the exact same thing. They're going to meet in the middle. In this case, you can see the middles are right over here. And then those individual chromosomes are going to line at the metaphase plate. Again, you can see that right over here again, right in the middle. Anaphase, remember, ana away, they're going to be pulling away from each other, just like this. And you can see that they're pulling away and away. Now, one thing I do want you to notice here is in anaphase 1, you notice that the chromosomes look like an X. In anaphase 2, those X's now are getting pulled apart. So you can see that we're reducing down further and further. Telophase cytokinesis, again, this is going to be end of the second nuclear division. So again, the exact same thing's going to happen again. So nuclear membrane reforms, cytoplasm is going to split, but we ended up with 4 haploid gametes. So each gamete is going to have one set of chromosomes, and all gametes are now going to be genetically different. So again, everything is going to be completely different because again, we're producing those sex cells and we want that genetic variability. We want things to be different. Now, I'm sure you might be asking why. Why is it so important for things to be different? And one of the big reasons why is because it's kind of an evolutionary advantage. If everything was exactly the same, and let's say that there was a virus, a bacteria, or something that can affect one person and cause death, if everybody is exactly the same, then we would all be vulnerable to that. But because we have that genetic variability and we're all different, we have that kind of survivability. So again, it is important to have that genetic difference when we're producing our gametes, because again, we don't want everything to be exactly the same. There are certain places and times where we do want things to be the same, like when we're producing more skin cells. But when it comes to our sex cells, when we end up producing offspring, when we have an egg and a sperm come together, again, we want that genetic difference because we want people to be different, which gives us that evolutionary advantage. All right, and that is going to conclude the first portion of this lecture. We're going to talk about non-disjunction in just a second, but what I'd like you to do is go ahead and take a second, pause this video, and answer these 3 questions. All right, and welcome back. So let's go ahead and check out these questions. So the cells produced by meiosis are genetically different. Remember, one of the major things is when it comes to meiosis, we're dealing with our sperm and egg. So we have our sperm and then we have our egg. We want all our sperm to be different. We want our eggs to be different because when they come together, we want a genetically unique and different person. Although they're going to share a lot of the genetic bases and genetic material from the parents, again, we do want them to be genetically different. So which of the following correctly describes meiosis? Meiosis begins with a diploid parent cell, and we're ending up with 4 haploid daughter cells. Again, the reason why is because we're going to have our meiosis 1, which we have our PMAT 1 and cytokinesis. And then we're going to go through meiosis 2 one more time, going through PMAT 2 plus cytokinesis. So we can end up with 1, 2, 3, 4 genetically different daughter cells. So again, we're going from our parent all the way down to our haploid cells. Which of the following correctly describes how mitosis and meiosis are different? Mitosis produces genetically identical daughter cells. Again, that's important because mitosis, just like my toes are part of my body, mitosis deals with our body cells. So we want genetically identical cells. If I damage my skin, or I'm growing, I want to produce more skin cells. I'm going to go ahead and divide this out into 2 identical. We want these to be exactly the same because we want to replace and we want to grow what we already have. So in this case, we have our diploid, or in this case, our 2N, because again, we want all of them to be the same. while meiosis is going to produce genetically unique daughter cells. Okay, and again, the reason why is because now we're dealing with our sex cells, which are going to be our sperm and our egg cells. And in this case, these are going to be haploid or just an N. Okay, and again, we have a lot of ways to make these unique. Remember, we have our independent assortment, and then we also have that crossing over. So again, we're going to have a lot of mixing and exchanging and changing of these. So these are now going to be genetically unique versus these are going to be genetically identical. So I'm sure you're wondering what can happen when meiosis doesn't go as planned. So this is going to give us something known as non-disjunction. So notice in meiosis, we have so many different processes we need to go through. Again, we have our meiosis 1, we have our meiosis 2, we have that crossing over, we have our independent assortment when they line up in metaphase. So we have a lot of things that we have to kind of watch and kind of keep track of. But there are times when things don't go exactly the way we want them to go. So this is essentially what non-disjunction is going to be. So this is going to occur when homologous chromosomes in meiosis 1 or sister chromatids in meiosis 2 fail to separate during meiosis. So normally, following meiosis, each gamete has one copy of each chromosome, resulting in non-disjunction. Or results of non-disjunction, gametes are either going to have too many or too few. So looking at this over here, you can see that this is our normal meiotic division. You can see in the beginning, we have this over here. You see we have 1, 2, 3, 4 to go through meiosis 1. Remember, they're going to meet in the middle. We have our microtubules or fibers that are going to pull these apart, again, separating them out. Now we're going to go ahead and have one over here, and we're going to have another one over here. So we have 2, and then these again are going to separate and go through meiosis 2, and then we're going to end up with 1, 2, 3, 4 gametes. Remember, these are all going to be genetically different. So these are all going to be haploid, and this is exactly what we want. But when it comes to non-disjunction in meiosis 1, you can see that they don't always separate out. When they don't separate out, again, you can see we have an issue in meiosis 1 where these are getting pulled to the left side and this one's getting pulled to the right. So now we're going to end up with 1, 2, 3 on this side and 1 on this side. Because of this, when we go through meiosis 2, again, it's going to separate out. You can see that separation. And now you can see we have 1, 2 that are going to be an n plus 1 because now we have way too many on this side. And then over here, you can see that we have n minus 1, meaning that we are missing right here. And because of this, this can also cause genetic disorders. Now, this is what it would look like when it comes to non-disjunction in meiosis 2. Again, you can see we have our starting point here. In this case, in meiosis 1, we have that splitting, and that split is perfectly fine, or that division. But again, you can see in meiosis 2, we have this part that didn't necessarily split apart, and this one did. So now we have one set that is exactly what we want. We have our N or a haploid versus this over here, because we have that non-disjunction, it didn't fully split apart. On this side, we have 1, 2, 3. And then this side, we have 1. And you can see over here, we have an n plus 1, and we have an n minus 1. And because this didn't split out completely, again, this can also cause genetic disorders down the line. All right, so let's go ahead and take a look at some of these review questions. So same thing, I'm going to go ahead and give everybody a couple of seconds to pause the video, give this a try, and then we'll check it out. All right, and welcome back. So let's go ahead and check these out. Crossing over is important because it allows for more genetic diversity in our sperm and egg cells. So again, when we have that crossing over, it's that exchange of genetic information. So it's increasing our genetic diversity or genetic variability when it comes to our gametes. Again, in meiosis, we do not want exactly the same. In mitosis, we do. When does genetic crossing over occur in meiosis? It's going to be in prophase 1. Metaphase of meiosis 1 and metaphase of meiosis 2 differ in what? Homologous chromosomes line up in meiosis 1 versus sister chromatids line up in meiosis 2. Remember, the reason why is because we're trying to go from our diploid down to our haploid. Meiosis 2 is similar to mitosis in that sister chromatids separate out during anaphase. So remember, ana is a way, so that means they're pulling or moving in opposite directions because they're going to split apart. Asexual reproduction, or reproducing organisms, produce offspring that are genetically identical to each other and to the parents. What type of cell division are the offspring a product of? In this case, it is going to be mitosis. In mitosis, you're going to have exactly the same organisms versus in meiosis, you have different ones. There are some organisms that do reproduce asexually, and they have some reasons as to why they go through this and have genetic identical offspring, or in this case, clones. But we won't really get into that too much. One of the major things to know is mitosis is going to be exactly the same. Meiosis is going to be different. When it comes to people, mitosis is going to be for your body. And then in this case, for meiosis, it's going to be for your sex cells. And we want that genetic variability to increase survivability. So there's going to be many advantages when it comes to that, but we won't really get into that today. All right, and then we have another set of questions. So same thing, go ahead and take a second, pause this, see if you can answer it, and then we'll go ahead and come back. All right, welcome back. Let's go ahead and check these out. Which of the following cells undergo meiosis? In this case, sperm and egg cells. Meiosis, remember, is going to be all about our sex cells. So it's going to be our sperm and our egg. Everything else, liver cells, skin cells, are going to go through mitosis. Which of the following is not a characteristic of meiosis? In this case, it functions to grow and repair the human body. Remember, this specifically is going to be for mitosis. Human egg and sperm cells are haploid. Remember, it's going to be an N, and each are going to have 23 chromosomes. So remember, haploid, H, represented by an N, is going to be 1/2 of diploid. Diploid is going to be 2N, and that is going to be twice the amount of haploid. True or false? It is likely that every egg a woman produces is unique due to crossing over and random assortment of chromosomes during meiosis. This is going to be true. Remember that crossing over event is going to increase genetic variability and independent or random assortment of our chromosomes during meiosis. All right, and just like that, that is going to complete this lecture. So again, if anybody has any questions, comments, or concerns, please feel free to reach out and I'd be more than happy to answer any of your questions. With that being said, I look forward to seeing everybody in the next lecture.